Good evening, everyone. I welcome you all to this meeting. Yeah. Good evening, everyone. Myself, Dr. Devin Gagar from Manalpha Med. It's my pleasure to welcome you all to today's session. Welcome to this webinar, topic named Innovator Semaglutide and the Current Market Search, What Clinicians Should Ask. As the burden of diabetes continues to rise, with a significant proportion of patients also facing weight-related challenges, diabetes has become a critical clinical concern. In this evolving landscape, innovator semaglutide has transformed the way we approach glycemic control and weight management. With the current market surge, it becomes increasingly important to understand what truly differentiates therapies from manufacturing expertise and clinical evidence to real-world experiences. Today's discussion aims to bring these perspectives into focus and support more informed, evidence-based clinical discussion. To take this forward, we are joined by our esteemed panel of experts, moderated by none other than Dr. Srirang Godbole, Director at Institute for Treatment and Research in Diabetes and Endocrinology, Dr. Ramesh Goyal, Senior Consultant and HOD at Department of Endocrinology and Diabetes at Apollo Hospital, Ahmedabad, and Dr. Samit Goshal, Consultant at Nightingale Hospital, Kolkata. Over to you, Dr. Srirang Godbole.
Thank you, everyone. Good evening. I'm extremely sorry for the technical glitch, due to which I'm sorry you're unable to see me. Nonetheless, we'll move forward because we have already wasted precious time. I think after 21st of March, there has been a flood of the so-called bioequivalent semaglutide in the Indian market. And that has led, of course, admittedly to increase accessibility. But what clinicians need to know is the science. I mean, the prudence, how to discriminate between the innovator molecule and the so-called bioequivalents. What is the status? Are they really the same? Are they all equal? This is something each of us as responsible clinicians ought to be well informed before we take decisions in the interests of our patients. Because finally, what we want is good patient outcomes. So, will serving the patients be possible by using the bioequivalents, or should we stick to the innovator? That is something which is of utmost importance, and that is what we'll be discussing today. I'll be just discussing a little about the peptide chemistry, which of course, is the backbone of semaglutide outcomes. Uh, yeah, this is something we all know, the huge burden of diabetes, which is largely fueled by the huge burden of obesity, generalized as well as abdominal. And more than 90% of people with diabetes probably also battle with weight issues. So this is something called diabesity and needs to be urgently addressed. And now we have molecules like semaglutide, which address these twin epidemics. Next. Uh, Basically, in the field of diabetes, it's the proteins and the peptides that have been in the forefront of not just diabetes care, but metabolic care. And of course, the most prominent proteins or peptides being the insulins historically, and more recently, the GLP-1 receptor agonists. And these really have been truly significant breakthroughs in the field of metabolic care. If you look at the history, if you can move to the next slide. If you look at the history, it's more than now 100 years when insulin was discovered by Banting and Best. And I think one thing that has been a regular feature for the last 100 years is the expertise of Novo Nordisk. And right from the isolation of insulin, and those of in the audience who are seniors will recall using animal insulin preparations by Novo. And then in the 70s came the recombinant human insulins. Then in the 90s, the rapid and the long-acting analog insulins, followed in the 2000s by various coagulation factors and recombinant factor 7A. All these technologies came into being in the 2000s. And then very, very significantly, what really caught the imagination of the medical world is the first peptide in a pill, the Rybelsus, the oral semaglutide, followed by Ozempic, which has caught so much of attention worldwide. It's a very consequential molecule. And in a higher dose, it's called Wegovy. So it's been a very illustrious journey along with Novo Nordisk, and they have been experts in this field of metabolic protein chemistry. Next slide. The recombinant DNA technology per se, Novo Nordisk has more than 40 years experience and expertise in that field. And I think what truly can be described as virtually a Nobel Prize winning, innovation is the world's first peptide in a pill. And that's going to, I think, open the floodgates for more peptides that we will see coming in a tablet form. This is truly a revolution. Next slide. You could move ahead. So what we are going to have today is a conversation. Don't move so fast.
So,
Uh,
sorry, sir,
yeah.
that was a glitch. Yeah.
Well, semaglutide, of course, has been in the Indian market, but it started off with the oral form, the Rybelsus, in 3, 7, and 14 milligrams. And then we had the introduction of the innovator molecule, the injectable semaglutide called Ozempic in doses of 0.25, zero point 0.5, and 1 milligram. And in those and in higher doses of 1.7 and 2.4 milligrams, known as Wegovy. So we both have now the injectable and oral formulations for convenience. Just move ahead as I speak. We know that oral Rybelsus is purely an anti-diabetic molecule, and the convenience is the oral route of administration. And the injectable, when you call it Ozempic, that's for early management of type 2 diabetes, and by which I mean even as a first-line therapy for diabetes. And then for purely chronic weight management, the molecule is called Wegovy. Next. So basically, we have these original biologics, the Rybelsus, the Ozempic, the Wegovy, and these have been innovator or originator molecules produced by a very complex biological process, the rDNA technology. But we also now have something called biosimilars, and we are used to that term in the context of biosimilar insulins, even biosimilar liraglutide, some brands were available. These are, of course, highly similar to the innovator, but they may have slight differences. The important thing is these are all produced by biological processes. And then you have synthetic copies, entirely, completely chemical processes. These synthetic copies are not necessarily identical to the innovator. And now the examples, of course, have started flooding the market after 21st of March. That is the injectable so-called bioequivalent semaglutide copies. Next slide. But are the synthetic injectable copies the same as the original Ozempic or Wegovy? You can copy a molecule, but can you copy the process which is used to produce Ozempic and Wegovy? Next. Remember, synthesis of proteins and peptides is a highly complex phenomenon. Next slide. And therefore, I think it's important to educate ourselves. And Dr. Samit Ghoshal will be precisely doing that. He will probably tell us now the science that should shape the clinician's preference and whether all semaglutides, are they really created equal? Dr. Samit, I look forward to listening to your lecture.
Dr. Samit,
Please,
you can take
please
in
take the
charge of the
dais.
screen.
Thank you, Dr. Godbole. And this is, I think, a very important juncture that we are at present, where we have a flurry of biosimilar products which have been approved. Now, here comes the interesting part. Once they're approved as biosimilars and it's there in public domain to be used by us at a lower cost, are they then not absolutely the same molecule that we have been using as the originator one? And that's a very interesting and important question to address. So I'll just go through a few important slides. The first thing we need to remember that biosimilar or creating the same molecule does not mean we have created the same medicine. It might be a very subtle difference, but a very important one. It is an assumption that with multiple semaglutide products, crossing the barrier of production and the manufacturing jargon and coming up with this molecule, matching exactly the 31 amino acid structure that they have with the original molecule, that they can be easily used interchangeably with the originator molecule. We need to remember that none of the modern molecules, or the ones which have come in as biosynthetic molecules or biosimilar molecules, they do not have the license of interchangeability. So that is the first important distinction we need to understand. The question is why? They have matched the molecule, which is true, especially the injectable one has actually matched the molecule. The oral one has not even matched the molecule. And here we need to distinguish even between the synthetics which are coming into the market as orals as well as injectables in comparison to the originator molecule. What we need to understand is that the process by which this molecule is made is completely different. The originator molecules are made from yeast cells. So there is this is a hybrid method. You have a biological process, and then you have a chemical process. Why is it important? Because at the end of it, whether we do it in a hybrid model or we do in the SPSS, which is a completely synthetic manufacturing of the product, which is called solid phase peptide synthesis, but in absolutely using precision technology and engineering, you just put one amino acid after another and create the exact 31 amino acid structure. So why the difference? Because at the end, in both these processes, whether you're using a hybrid one or you're using the chemical SPPS method, you have the same 31 amino acids that you're manufacturing. So why bother about it? The problem is these are not linear molecules. These are not that you put in amino acids one by one and put them in a straight line. These are protein structures which, after being assembled, folds. And the folding can be of two folds. One, it can be a secondary structure and a tertiary structure. Secondary structures are pleated structures, and tertiary structures are in three-dimensional space. Now, this is something which is extremely important, because even after having created the 31 amino acid in a linear fashion, do they fold in the appropriate way? Because that is going to be a critical determinant of its pharmacokinetic and pharmacodynamic potential. What we can see is what do the regulatory bodies have to say? So as clinicians, whenever a biosimilar company comes and meets us, as I have started asking them these questions, these are the four important questions we need to ask them. These questions are true irrespective of whichever regulatory bodies are assessing the biosimilars in comparison to the originator molecules. This is the Indian one, CDSCO, and they have an orthogonal way of assessing this product. FDA has slightly different association. Again, we need to understand why we are saying this, because a few of the pharmaceutical companies are telling us that it is not only the biosimilar semaglutide which is available in India, but it will also be exported abroad in the BRICS country, maybe in Canada and others, where obviously not necessarily the CDSCO guidelines, but also the international guidelines like the FDA recommendations or the EMA recommendations will become important. Here we need to understand that there are differences between the different bodies or regulatory bodies as their requirements for the pharmaceutical industry to qualify as biosimilars. So these are the four important questions, because these are the four important attributes a company has to qualify before they can apply their molecule as biosimilar. One, and most important, probably more than 90% of the requirement of all regulatory authorities is to do a very, very good quality structured analytical similarity test, which means you have to prove the primary structure, which is the 31 amino acid, the secondary structure, which is the folding of the peptides in 2D space, and the three-dimensional or tertiary structures in the 3D space. Now, as far as CDSCO is concerned, a qualitative assessment is good enough. I'll show you FDA needs a quantitative statistical analysis of it. Now, having proven that it's the same analytical structure, you have to do an absorption study. Are the originator or the biosimilar molecule absorbed post-injection in a very similar manner? Three attributes important. One, the maximum concentration reached in plasma called the Cmax. And two, area under the curve. One is area under the curve from 0 to the time of which the last sample was drawn for assessment. And area under curve from 0 to infinity. Infinity means when the entire drug is washed out of the system. Do they happen? So is the maximum concentration peaking between the originator as well as the biosimilar molecule at the same time? And are they getting washed out of the system at the same time? That's the similarity we're looking at. Third is the pharmacodynamic similarity. Now, testing clinically for equivalence as far as HbA1c is concerned, weight reduction is concerned, is actually optional. The most important tests are the first two and the fourth one. This is an optional step. Unfortunately, we are seeing majority in the ad boards, this is the one which we have been shown that, see, we are reducing HbA1c and the weight practically equally compared to the other molecule. That is not the focus of the regulatory bodies. That is not the focus of us clinicians. Our focus is whether you have a similar molecule and at lesser amount of impurities. Now, assessment of impurities are also different, and we need to understand why. Now, since we have a biological process from yeast, there are different types of impurities which are collected during that process. Now, whether you have got rid of it, we need to analyze them. But in the chemical process, the impurities are completely different. So by just saying that drug antibody is same between the two is not good enough. The process-related impurities are different and they need to be analyzed in a differential fashion. Now, I will not name, but these are the two the biosimilar companies. And when we go through their data, we find that there are differences in which the different biosimilar data, even though they need to go through all those 4 strategies that CDSCO has laid down, but there's a lot of difference between the way they have presented their data. And I'll show you a few of them, the differences and similarities. The first one have shown that there is a near 100% pharmacokinetic exposure, which is fantastic. 0% anti-drug antibody incidence. And they have done a deep, broad, and orthogonal analysis. But remember, the statistical parts are missing. They're mostly qualitative. Cons, in the same one, when the pharmacokinetic studies were done, the 3 pillars for PK studies are Cmax, area under the curve 0 to the hours when the last sample was drawn, and 0 to infinity. Infinity means when the washout has happened. The AUC 0 to infinity data is missing. Secondly, the statistical analytic framework for the analytical studies, which is that you have to have your data within 80% to 125% spread. That is the upper and lower limit is again missing. Surprisingly, in the bioequivalence study, there is a statistically significant larger HbA1c reduction in the biosimilar one compared to the original molecule. Now, if I'm claiming for bioequivalence, how can I say that I have a statistically significant larger A1C drop? Now, these are important question marks which are coming from this study, but maybe they were not that important enough because the clinical studies were optional and hence probably they were not given that amount of weightage when the approval were given. The second biosimilar company, when they presented their data in their ad board, showed a perfect HbA1c equivalence, a complete pharmacokinetic data set, detailed safety analysis. Again, the statistical analysis part in the analytical assessment was missing. It is extremely important to understand that you need to match the different batches. So it's the batch-to-batch variability within the biosimilar ones and batch-to-batch variability within the parent originator molecule needs to be compared and presented in a statistical manner. So there is mention of batch 14 batch or 16 batch analysis, but the statistical analysis part is missing. None of the data have shown us anything about the neutralizing antibody, because if the neutralizing antibody sits on the receptor, then the original molecule will not find space and there will be blunting of effect. Unfortunately, it is not mandatory from the Indian regulatory body to present them, and hence probably it was not done. So we cannot force someone to do it. But having said that, this is something which is required if you want to get an FDA or EMA approval and then market it into the other part of the world. So this is the bridge. CDSCO, the recommendations in India have been met by the Indian biosimilar companies, and hence they're approved and they're in market. But if you're going to go abroad and try to put this molecule across in the broader scenario, exporting in other countries, then these are the 3 important areas which have not been detailed or presented in our ad boards. and they are missing. Maybe they are there. Maybe those data were not shared, but probably it will come out of the bag prior to the international launch. We don't know, but has not been shared as of now. The biggest glaring, I think, issue is not the injectable one, because they have matched the 31 amino acids perfectly, but it's the oral one. And the reason I'm saying this is if you look at the oral one, as I said, injectable one, the biosimilars have exactly the 31 amino acids in the same sequence as the originator one. Here, the oral absorption, in order to make it, the delivery system which was used is SNAC. And now something else is added to the original semaglutide molecule to help it into absorption. This is completely different from the one that we are going to have in the market in the oral semaglutide biosimilars. Remember here, it is something else. So if you have semaglutide plus A, now we're going to have semaglutide plus B, making both of them two different molecules. Not only that, their absorption is also completely different. The innovator molecule, which is Rybelsus, their absorption is completely transcellular. The new one, using a new system called the PE system, has both transcellular and intracellular absorption. This is definitely going to have an impact on the pharmacokinetic variability. So we cannot call them the same molecule. Unlike the injectable, where they are the same molecule, and there are certain nitty-gritties here and there which needs to be taken care of, it is not so when it comes to the oral molecule. So this is what we can say. Since the molecule is different, its absorption is different, just by doing some bioequivalent study, which is optional, and showing that we have equivalent A1C or weight reduction, does not translate into outcomes benefit. We might do away with outcomes trial with injectables. We cannot do away with outcomes trial with the oral drug. We cannot say you have a heart disease, you have ASCVD, and you have a type 2 diabetic patient requiring a GLP-1, and I'm giving you the dual PE-based semaglutide, you will get the benefit. This inference cannot be done. So what we are getting in the orals one are not biosimilars. They can be termed as follow-on biologics, where the exact data accumulated on outcomes from the originator molecule cannot be extrapolated on them. So the danger is that there is an overemphasis whenever in the ad boards we come to see that they just skip over the analytical part and overemphasis on HbA1c and weight equivalence. No, that is not the lookout. That's just the tip of the iceberg. The major part that needs to be focused on is what exactly are the analytical process you have done? What are the impurities that you're aggregating? Surprising from the side effect point of view, gallbladder stone, pancreatitis are also missing as signals or side effects from most of the data which are being presented. So I can summarize by saying that although, especially with the oral ones, there are glaring differences that we can see. We will see that most of the time, the data which will be presented will be heavily dependent on the bioequivalence part. Again, I'm stressing on the point, it's the optional part, optional requirement of the regulatory authorities. We cannot extrapolate the originator Rybelsus molecules cardiovascular data with the one oral that we are going to have in the market. We cannot extrapolate the renal data onto the oral one that we're going to have in the market. We are not expecting the same PK studies because of different absorption profile as documented by the companies who have brought the molecule themselves. So what I would love to propose as a hypothesis algorithm. Now, these are approved molecules. We cannot say don't write them. They are approved by our regulatory authorities. They will be in market. There are cost benefits. So we cannot deny our benefits from cost benefits. But I think if the patient presents with established ASCVD, having a PTC, had undergone a CABG, has an established heart failure CKD, and there is a need for long-term multi-organ outcomes protection, I think the first thing that we need to discuss of a patient is the innovator is the drug of choice. Cost coming in, we can think of the injectable, definitely not the oral counterpart. Simple type 2 diabetes management, simple glycemic control, there's no outcomes which is involved in the backdrop. We can think of using the biosimilar injectables. Again, I'm saying not the oral one. Now, not all semaglutide products are identical. Their manufacturing is different. We're seeing from the oral part, their absorption is different. Their pharmacodynamics are different. Some of the biosimilar companies are showing they have superior A1C lowering. So a lot of difference are coming in. And we need to understand that the local regulatory approval does not equal global biosimilarity approval. As I showed you, that they ours is a little bit more relaxed than the FDA EMA. So once you go for those additional questions will definitely be asked, and that's important to see how they are responded to. In high-risk patients, we prescribe evidence. In low-risk patients, we can prescribe equivalence. Real-world cost determines whether treatment happens at all. So even in some patients where we know they have ASCVD, but due to cost constraints, we can't give them the originator molecule. I still think there is no harm in giving them the injectable biosimilar that we have. So with this, I'd like to end my presentation and hand over to Dr. Tere. Thank you very much.
Thank you so much, Dr. Samit. That was a wonderful lecture. I think it clearly and scientifically put forth the various pitfalls in the use of the so-called bioequivalent semaglutide. And you said there are patients of high risk and low risk. I believe most, if not all, diabetic patients are high risk, So I would rather use an innovator. I always put myself in the patient's place. What if I were the patient? Would I use an innovator semaglutide, or would I use one of those copies? And I think the answer for myself is clear. I would go with the innovator. I would certainly not use a copy for myself, unless, of course, I'm desperately poor and I need something, and something is better than nothing, then probably I would go for the copy. But if given a choice, there's no question. All the outcomes are on the innovator, and you mentioned so many of the manufacturing details and the regulatory, pitfalls, which most companies probably would falter at if they were to try to export their molecule abroad. That was a wonderful lecture. But now we also have Dr. Ramesh Goyal in our panel, and we will have a small conversation on the relevance of expertise, the evidence, and the experience with the two very esteemed panelists, one of which, of course, is Dr. Samit. Yeah, next slide. So first, Dr. Ramesh Goyal, welcome to you. Dr. Ramesh Goyal is from Ahmedabad. He's a very eminent endocrinologist. Uh, well, we were discussing recombinant DNA technology, and what is being followed in the copies is a chemical process, just assembling 31 amino acids, as Dr. Samit put forth. So, how does our DNA technology positively impact the final semaglutide end product, Dr. Ramesh?
Yeah, so when we talk about technology, and particularly when we are talking about peptides, the technique by which this end product is product is formed is very important. So, recombinant rDNA technologies. Am I audible?
Yes, yes, you are. Yes, please go
Okay.
ahead.
So there was some confusion. So when we are talking about biosynthesis of peptides and proteins, the process by which it is manufactured is very important. And recombinant rDNA technology is a time-tested platform to manufacture proteins and peptides. So there is a gene of interest identified and cloned. So I'm describing this process, and then clone gene into DNA vector is incorporated, and then there is transfer of this plasmid into host cell, and cell culture or fermentation process is done, and then there is downstream processing, and then there is a purification of the drug substance, and finally, after you can see all these steps, there is formation of the product for the use. So it is a highly complex process, and the process defines the final product when peptides are involved, as I mentioned earlier. So when we talk about this time-tested recombinant DNA process, it ensures that there are low peptide fibrillations, then there is high purity, and there is low immunogenicity, and there is assured consistency, and there is a persistent quality, and there is a proven safety. So I think everybody will agree here that all these parameters are very important when we are discussing about molecules like insulins and GLP-1 RAs. So all of this six factor, they are most important.
Dr. Samit, you did mention some, details about manufacturing, but really do these differences in manufacturing, the process itself, does that impact clinical outcomes? Because there are no clinical, studies to tell us what happens if you use a copy. You have study with the innovator, but you don't have a study with the other molecule. So, I mean, would these differences make a difference?
Yeah, so I think mostly you will see that the biological company, the biosimilar companies will show you clinical data restricted only to HbA1c and weight. Now, as we know that the cardiovascular benefits are irrespective of glucose control. Today, at least we know that. So by just saying that the glucose control was similar between the two, can we then extrapolate a SOL data or a flow data or a stride data and say, okay, it's the same out here? It's very difficult, and probably not. Second part is the different manufacturing processes, including the way that the molecules are stored inside the vials or inside the pen with preservatives like phenol, can create a lot of aggregates. If you remember those days of erythropoietin where we reported a death, and that was reported in NEJM because of aggregates, protein aggregates formed from biosimilars, with a lot of them being even taken out of circulation, including a Marvel insulin taken out of circulation, even though they were biosimilar from glucose lowering point of view. So it's extremely important to understand the long-term impurity related side effects, because only thing that has been assessed is anti-drug antibody, and that is zero in some 0.68 in some percent in some of this data. How this is going to pan out in the long run, we don't know. I think the phase 4 pharmacovigilance is extremely important from the side effect perspective. I still say that outcomes benefit, I think we still have to rely more on the innovator molecules. And as you rightly said at the end of my presentation, that an extremely poor gentleman who could not take GLP-1 because of cost, definitely we can try them on this molecules. Some treatment is better than nothing. But then I think I'll just be looking in the next 2 or 3 years on how the adverse events of these patients are progressing rather than the effects per se.
Right. And I think you made a very fine distinction between injectable and oral semaglutide. That really makes a very big difference. The snack, how much you use it and the type of snack. Because the innovator snack actually underwent a CV outcome trial. Many people don't know about it. So none of these molecules, I believe, their whatever absorption enhancer, I don't think has met those rigorous criteria. So now can we ask the next question to Dr. Ramesh Goel? Again, something was discussed about the regulatory, approval differences in India, in the US FDA and the EMA and, other countries. So do these synthetic copies come with the same regulatory approvals as with innovator semaglutide? Obviously not, but could you just throw some light on that?
Yeah.
We have local
So
approvals, but
yeah. So the innovator semaglutide has wide indications and wide approvals, because obviously the innovator semaglutide has undergone all the clinical trials related to type 2 diabetes, obesity. Then we have trials on NFLD and MEFAL, and we have trials for cardiovascular outcomes. So there are broad range of on-label clinical indication for Ozempic in India. So once weekly Ozempic semaglutide injection is approved primarily for first line management of diabetes as an adjunct to diet and exercise to improve glycemic control in adults with type 2 diabetes mellitus, and additionally to reduce the risk of major adverse cardiovascular events in adults with type 2 diabetes and established cardiovascular disease, and additionally to reduce the risk of sustained EGFR decline, end stage kidney disease, and cardiovascular death in adults with type 2 diabetes mellitus and chronic kidney disease. So apart from management of type 2 diabetes, Ozempic has cardiovascular as well as renal indications also. When we talk about Wegovy in India, so once weekly Wegovy injection 2.5 milligram as a primary indication for weight management in adults as an adjunct to reduce calorie and increase physical activity for weight management. Additionally, to reduce the risk of major adverse cardiovascular events in adults with obesity or overweight and established cardiovascular disease. And the approval awaited is to treat adults with metabolic dysfunction associated steatohepatitis with moderate to advanced liver scarring, that is fibrosis. So it is already approved globally for this indication and will soon get this indication for from our regulators also. Now, when we talk about Ribelcus, the oral semaglutide, the primary indication is an adjunct to diet and exercise to improve glycemic control in adults with type 2 diabetes, and additionally to reduce the risk of major adverse cardiovascular events in adults with type 2 diabetes who are at high risk of these events. And it is the only OAD to have an label indication for primary cardiovascular disease prevention. Now, when we talk about synthetic copies, they have limited regulatory approvals. So, one of the the primary approval is to improve glycemic control in adults with type 2 diabetes, used alongside diet and exercise in combination with other anti-diabetic agents or as monotherapy if metformin is unsuitable. And for few copies, it is indicated for chronic weight management in adults as an adjunct to reduce calorie diet and increase physical activity for weight management. So they do not possess on-label indication for long-term benefits, that is cardiovascular disease, CKD, MASH, etc. So only primary approval for majority of copies is for management of type 2 diabetes and for some copies for weight management.
Yeah, it's very nice that you made that distinction, because these companies, they just give the indications as if, they are manufacturing the innovator, which is not true. And those additional indications are actually what I feel are most important because they deal with hard outcomes. So that is what is important actually. Dr. Samit, what is the level of clinical evidence, both, RCTs, the real world evidence with innovator semaglutide? What sort of track record does the innovator have?
Oh, we see, semaglutide, I think, has the data is unparalleled. I mean, it is, you have data with as monotherapy, you have data in comparison to insulin, in comparison to other GLP-1 like dulaglutide and others. So the data is huge. And also you have the data in different outcomes, which is extremely important. It is the drug of choice now practically for MSLD. It is, a drug even surprisingly, one of the drugs for osteoarthritis, improving your walking capacity and reduction of pain. So the amount of indication that this drug has is enormous. Hereby comes the importance of if the indications are so large, so you have a very larger population who will be probably eligible for this drug. And hence this important question, how do I select between this and the biosimilar drugs, which have actually gone to do a, say, a 36 days or a 24 weeks just comparative study and has nothing else. But by virtue of piggybacking the data which has been done by the originator molecule, claiming all of these in their package insert, sometimes it looks grossly exaggerated. I think, yes, they should be allowed to be in market as biosimilar product, but piggybacking this entire data and saying that you're going to extrapolate them, I think is stretching it to a great extent, including there are only 5%, 10%, 11% of Indian or Asian data from them. Even if you pull them together, that data from the originator molecule is way bigger than whatever little bit is accumulated by most of the biosimilar molecules that we have.
Right. That's extremely important. When we present, I think, when we speak with our patients, we should make them aware. It's, I think, our duty to make patients aware what they are in for if they choose a copy over an innovator. They must know exactly the science, and the evidence. Dr. Ramesh Goel, cold chain is so very important, especially in a country like ours. Even device convenience is important for patient compliance and adherence. So, how would you rate the importance of these two factors, the cold chain compliance and the device convenience? Do you have enough confidence in the copies that they will be able to maintain the cold chain compliance and their devices?
So, Dr. Sheena, when we are talking about peptide molecule, for me, cold chain maintenance in a country like ours, where the temperature can go up to extremes, it is very important because for a drug to be effective, a cold chain has to be effectively maintained. So, a robust cold chain compliance is essential for Indian summers where the temperatures they touch between 40 to 45 degrees in most of the part of India. So, semaglutide being a peptide is temperature sensitive and requires a strict 2 to 8 degree centigrade control to preserve and peptide integrity and clinical potency. So, innovator semaglutide comes with the assurance of end-to-end cold chain compliance from factory to fridge. And we all know that Novo is dealing with insulin, and we have been using Novo insulin since long. And we all know that the cold chain assurance is there when we are using a Novo product. Now, device is integral part of injectable semaglutide therapy. So, innovator semaglutide comes in a simple to use flex touch pen. So, and it comes with a 4 co-packaged no fine plus needles. So there is, we all know we have used this flex touch before also with products like Rhizodrug and Tresiba, and there is no push button extension. There is low injection force, and it is easy to use, and confidence in drug delivery, and 100% patients they recommended flex touch in clinical trials.
Dr. Samit, so many patients would be coming to you, I'm sure, and say that they have read in the newspapers and in the media that so many copies are now available at a cheaper price, and they ask you to switch from the innovator to a copy. So, do you see there is a clinical need to do that?
I think we have discussed from our ends that we are definitely going to tell them the pros and cons of doing, changing over to biosimilars. The issue is if the patient still decides to take the low cost one, I think the decision ultimately lies with the patients. But it all depends on how we place the data in front of our patients. Like, this is it. They're in market for so many years with this data. The copy has come. The choice is yours. And if someone really wants to, the question is, do we really stop them from having one? I think that's something we can't do. So, but secondly, let's be very clear here. Someone who's already on the innovative molecule, these they have definitely, the regulatory bodies have made it have ensured that these are not interchangeable products. You cannot use them interchangeably. So if someone whom I am initiating, definitely all those questions will be discussed. Someone who's already on and wants to go to the lower product, we have to tell them that they are not interchangeable, but the final call definitely lies with the patient. But they are definitely not interchangeable. As this slide shows that two-way crossover trials are required to prove interchangeability. But again, there is a problem with this slide because with a long-acting agent like semaglutide, you cannot do a crossover study because it will take months to complete, because the washout period will be 5 half-lives, which will be few weeks in between. So it is not recommended to do crossover trials. Parallel group trials done by the biosimilars are absolutely fine. So there's nothing wrong with it, except if you want to have a complete interchangeability data, you probably need a clamp study to back you up, which are not there in these trials. So they are not interchangeable. We have to again tell our patients they are not interchangeable. But if you want to switch for cost purpose, the decision has to come from the other side of the table. It is not. It should not come from our side.
So, only today I had 3 patients coming to me in person and a fourth one writing an email to me saying that they read about, these copies and they had come fully prepared, expecting that I would straight away change them from the innovator to the copy. And when I placed the data before them and when I made these points, which were really discussed in the course of this webinar, surprisingly, I was also surprised. They said, no, we'll choose to remain with the original. So patients who can afford, if given the proper data, will, I'm sure, opt for the original. Who doesn't like the original? I mean, who likes to go for a copy unless there are financial constraints to, choose a copy, which I can fully understand. But really, finally, it depends on the doctor, how they place this data, which Dr. Samit and Dr. Ramesh Goel placed before us. I think it added to our wisdom, I think. So finally, why are synthetic copies not the same as the innovator? And I think this was clearly explained in the wonderful lecture which Dr. Samit said. But if you just have to summarize in one slide, here it is. Recombinant DNA semaglutide, the innovator, has a track record at least since 2017, whereas synthetic have been just introduced maybe a week ago. And the technology is entirely different, the process is different, and see the track record of the innovator, more than 50 clinical trials, more than 60,000 participants. You can't hold a candle to the sun when you talk of synthetic semaglutide. The regulatory approval for the innovator, of course, is global. More than 75 plus countries have given their approval, whereas for synthetic, it's just a local approval. And with Novo, I can tell you from my experience of more than 40 years, I'm very confident about their cold chain. You can't dispute that. I'm not so sure about some of the companies that manufacture the synthetic ones. I've had bad experience with some of their insulins in the past. And the device, as we know, Flex Touch, we're very, very familiar with that. And the patient experience is more than like 49 million patient years. All of these points are missing with the synthetic semaglutide. So in terms of quality, purity, the consistency, and the cold chain, and not to speak of the device, I think if given a choice, and if I can afford, no question about it, one would always prefer the innovator. And if only there are financial constraints and nothing else can be given, then only I think one should think of synthetic semaglutide. Next slide. Of course, I think this is all well known. We start always with typically 0.25 milligrams for the subcutaneous variety, and then typically escalate the dose every 4 weeks, but we can go slower if we want to. And we can also de-escalate if there is a patient need in terms of side effects. And with Wegovy, of course, you can move on to higher doses of 1.7 milligrams and 2.4 milligrams. With Ozempic, the ceiling dose for type 2 diabetes is 1 milligram once a week. Oral Rybelsus typically start with 3 milligrams. After 1 month, move on to 7 milligrams. If required, then at least after 1 month has elapsed, you may choose to go up to 14 milligrams if you need it. So this is how you typically dose the innovator semaglutide. Next slide. I think this was a wonderful discussion. It threw a lot of light, and the noise that is there surrounding synthetic semaglutide. Clearly, some sense has to be introduced into this cacophony of synthetic semaglutide. I think the two panelists, the faculty, Dr. Samit and Dr. Ramesh Goel, did a wonderful job. Thank you for having me here. It was a pleasure to listen to this faculty. Dr. Devan, would you like to now take over? Thank you so much.
Yeah, so the audience has some of questions for you all.
I can't see any questions in the chat box.
Yeah. So it was a live session. So they have
Okay.
that's a different chat box actually.
Right.
The questions are with me. So I'll just say the questions. So the first question is, when patients ask whether all semaglutide options are basically the same, how do you explain the difference in a simple but scientifically accurate way?
Well, Dr. Samit, would you like to take that back? It's just a summary of the lecture that you gave.
Yeah. So I think that's the simple thing is that like any molecule, you have the parent molecule and then you have like the generics after patent expiry. You have a very similar trajectory out here. So I just tell my patients that this is the original molecule, which I have been using for so long, which is in market with so much of data. Now the low cost biosimilars are available. but without the carrying the piggybacking on the data done by the rest. So if there are no financial constraints, it's better to stick on to the original products. That's what I normally tell them. But I agree with Dr. Godbole. Once you put it on table, you'll be surprised that a large number of patients who initially comes with cutouts really go back by themselves saying, no, doc, I think we'll stick on with the original product.
Yeah. So we have one more question from the audience. So they have asked if you all can share your real world experience on Wegovy, Ozempic, and Rybelsus for weight loss, HbA1c, or others. Dr. Godwal, if you can answer this question.
Well, I think these molecules are game changers, you know. I would say that they are the most consequential molecules maybe over the last 60, 70 years, after statins to some extent and SGLT2 inhibitors. But even more than those two molecules, I think it's the semaglutide amongst the GLP-1 receptor agonists. And even you can say even amongst, you count the twincretins also. They also do not have the outcome data as of now. So the only molecule really to have data across different organ systems and across different aspects of chronic disease really is semaglutide today. And therefore, we are not just talking of glycemic control or weight control. We are talking of cardio-metabolic, renal, hepatic outcomes when we are using semaglutide. So it's a disease-modifying, life-saving drug, I would say. All-cause mortality has been shown to be reduced. So, I mean, what else can you expect from a drug? And with so much of evidence in its favor, so many patient years, millions of patient years experience with semaglutide, I think there will be few patients who do not need a semaglutide today if you have type 2 diabetes. Most patients would be high risk. Most really probably would qualify for the use of semaglutide, whether it's oral or injectable. So I think we should place all this data before our patients, remembering that injectable semaglutide is a first-line treatment, by the Indian regulatory authorities. It's not a drug to be used as a third line or fourth line. It's a first-line drug, and that should be emphasized, I think. Thank you. It was an excellent discussion.
Yeah. So one more question, one last question actually. So the last question is, how does biosimilar differ in effectiveness from the originator molecule?
Well, I think this was discussed. What they merely put forth is the A1C lowering and the weight lowering, but these are soft outcomes. Finally, why do we want to lower the A1C and why do we want to lower the weight? These are, means to achieve good health. But finally, what matters are hard outcomes. What matters is your 3-point MACE, it's your myocardial infarction, it's your stroke, it's your MASH, it's your progression to cirrhosis, it is your EGFR decline. And where's the data for the synthetics? All the data that you have is for the innovator, which is why the innovator has those labels, which the synthetics do not have. So it's as simple as that, So if you go by the evidence, you use the innovator because that has the evidence for the hard endpoints, not just the soft endpoints.
That's all for today, actually. I sincerely thank you, Dr. Godbole, Dr. Samit, and Dr. Ramesh for your presence for today's webinar. And thank you, everyone. Thank you, audience, for seeing and for coming for this webinar, for giving your precious time. Thank you so much.
Thanks.
you.
Thank you.
Good night.
Thank you.
Good night.
Thank you. Good
Thank
night.
you.
Right.